Good afternoon, everyone. It's Shay again back here with another video. Um, this first video is going to introduce anatomy and physiology, and it's going to kind of dive into the different things like homeostasis, sensor stimuli, uh, stimulus sensors, integrators, and effectors. We'll kind of dive into the PowerPoint right now. This will just be a brief overview for when you come to class. So first of all, this is anatomy and physiology, and we need to kind of define what anatomy and physiology is. I can remember before I went into medical school, I remember having a gross anatomy class. That was going to be the first class that I was going to take that semester, and everybody had talked about how difficult the course was because you get a bunch of foreign terms. Because anatomy is really founded on this Greek and Latin um, uh, type of word. Uh, Foundation, and it's difficult at times. So you have to really break the word down. You'll see a lot of patterns. Ology is going to mean study of. So cardiology. So it's going to be the study of the heart. Uh, you're going to see things like that, and you're going to see patterns. And this may take some time, but I don't want you guys to be intimidated by any means because this is a learning process, and this is a great introduction course to things that you guys are going to be studying later on, whether you go into nursing or you decide to go to medical school or pharmacy school. This is just going to be a great introduction to kind of become familiar with these things. So again, like I said, anatomy is Greek and it means to cut open. So uh, previously, back in the older times when Aristotle and people of that sort were around, there was a type of medicine that was being practiced, and the medicine that was being practiced was more based on magic or some type of superstition, and it was more traditional medicine in that sense. They were not allowed to study dead bodies or anything of that sort. And our knowledge of medicine grew substantially once we started studying dead people. And I know that may sound crazy, but we had no idea what the inside of the human body looked like. In medical school, we had cadavers. What a cadaver is that's just somebody who's donated their body to science. So we would uh, because of these great people, we would be able to uh, study what the normal anatomy looks like. So, we would go through dissections, cutting open, whether it be the abdomen, and studying things like the stomach, the small intestines, things of that sort. So, again, anatomy is it's not changing for the most part. There are some anatomy that may differ from regular anatomy, but this course is meant to focus on what is normal anatomy. What is the normal anatomy of the human body? And we'll kind of go over the different structures of within this course, you'll go over the the musculoskeletal anatomy. So we'll go over different muscles within the body, and if you're going to physical therapy school or PTA school, that'll be very beneficial as you'll be using these muscles and all the time. And whether you're helping patients who have recovered from strokes or that whatever it may be, you're going to be talking to these patients, and you're going to need to know the function of the muscle. And when we start getting into the function, we start discussing the physiology, right? So anatomy is the Greek word for structure, but physiology, this is a different word. Physiology means the function. So let's use the heart for example. The heart is made up of three different layers. You've got the pericardium, which is the outside, the endocardium, which is the inside, and you've got the myocardium, which is kind of there in the middle. So that would be the anatomical structure of the heart. But all of those different layers may have different functional purposes of the heart. And what is the overall function of the heart? Well, the heart is responsible for taking blood, sending it out into the body right after it's received oxygenation from the lungs. So, again, we're talking about the physiology of the heart. So, its purpose, its biochemical function of living organisms and their systems. So, we discuss the anatomy of the heart, the different layers, and then we discuss the physiology. So, how does the heart work? That's just kind of a brief overview of what anatomy and physiology is. So, Um, Usually, you'll see a common pattern within this class. You'll see that whatever system we're dealing with, we'll discuss the anatomy first. We'll discuss how is it made up, what is it made up of, how is it made up of those things, and then we'll say what it does. We'll kind of go in that type of order. I feel like that's probably the best way to learn. So before we get into anatomy and physiology, let's kind of get the course breakdown and see where we're going to be at this semester. This semester is going to consist of primarily three different types of systems. So we're going to first begin with more so of a review. We're going to go over the normal biological and cellular structures and the physiology of those things. And this is just needed for foundational purposes. So everything that we will discuss is going to be built primarily on these principles. From there, we'll move into things like the tissues. Tissues are just going to be groups of cells, and we'll go into something called the integumentary system. That's just a fancy $3 word for systems dealing with the skin and outer tissues of that sort. So, and then from there, we'll go into the skeletal and to the muscular system. Those kind of go hand in hand. And then from there, we'll go into the nervous system. So, we'll have 3 exams throughout the semester. You can look in the syllabus and see that. Then we'll have a final exam. And I want to kind of reiterate this point that all of the concepts kind of build on each other. There is no cumulative exam, but it's important to have a good foundational understanding of the principles that will be introduced in each chapter, as they will build on each other. And then going here, this is just kind of an outline, just showing the body as a whole, how you have a different skeletal system, how you'll have the axial skeletal system, which is going to be primarily your vertebral and long bones, and then you'll have your appendicular, which will be which will be your appendages. Uh, then once of course, you have muscles that attach to these bones through different ligaments and tendons of that sort. And just giving kind of a brief overview of the nervous system, which we'll get into later on, dealing with the central and the peripheral nervous system. So before we get into all that, we first need to talk about the levels of cellular organization. And if you took my Bio 112 class, I kind of definitely made this a point of emphasis. Um, it's actually one of the traits of one of the characteristics of life, right? Life is organized. So this organization begins at a chemical level, right? We have atoms that we've discussed in kind of our um, chemistry portion of our biology. So atoms come together to form molecules, and the molecules will join together through monomers and polymers to form macromolecules. But essentially, at the cellular level, cells are made up of molecules, right? Does anybody know what the basic unit of life is? A Bio 112 class should. Exactly, it's a cell. The cell is a basic unit of life, right? And we know that groups of cells will come together to form something called a tissue. So tissue is uh, just a word for a group of cells. And then we know that tissues come together to form organs, and then organs come together to form organ systems. I love to use the example of the myocardial cell, right? So myo means what? Muscle. Cardio means heart. So myocardial cell. Well, it's the heart muscle cell, right? So myocardial cells come together to form myocardial tissue. Myocardial tissue comes together to form the myocardium. And what is the myocardium? That's the heart, right? So the heart, along with other organs such as the aorta, the coronary arteries, um, that forms the circulatory system or the cardiac system. And as you can see, things considerably get more complex the further we go up from the cell to the tissue, from the tissue to the organ, from the organ to the organ system. I think at this point you guys are probably aware of the levels of organization, but again, it's just a good review point. So again, is our body system maintained? Uh, our body system is maintained through a process called homeostasis. Homeo meaning same, stasis. So stasis meaning standing still. So our body is in constant fluctuations, whether that be electrolyte or whether that be through temperature. If it's cold outside, our body's going to vasoconstrict in order to conserve heat. Whatever that may be, our body is constantly trying to maintain this equilibrium or this balance. Because if our body shifts in either, if our body shifts um, in either, if our body shifts in um, just one direction, that's never good, right? If our body gets too hot, we can go into hyperthermia. If our body shifts too cold, we can go into something called hypothermia. So our body has built-in mechanisms in order to maintain a central set point. And as we'll discuss later on in this next portion, you're going to see that that is called an integrator. So we'll go over these key basic terms, and they're going to be responsible for kind of forming this regulatory process. So you're going to have a stimulus, and a stimulus is just going to be something that triggers a response, right? This is just going to be something that is interpreted by a receptor or a sensor. And the sensor is then going to send it to the integrator, or um, kind of like the guide control. He sets the set point, right? He tells our body what um, basically how to react, and then from there we'll get to the effector. The example that I'm going to use is going to be pain. So, pain, you're going to have a stimulus, such as, let's say you're cooking dinner and you're using a really sharp knife and you prick your finger. The blade comes in and pricks your finger. That's going to be the stimulus, right? That's going to be the input into the system. What's going to trigger these events. And from there, the free nerve endings, which are going to be present within your skin, that is going to trigger um, the integrator, the brain, to process this pain phenomenon. And then from there, it's going to send a message to your hand. You may grab your hand in pain. You may feel pain just in general, and then you're going to have a response. What will the response be, right? We may jerk our hand away. We may grab our finger, whatever that may be. So I just want you to see that this exists. You're going to have a stimulus. A stimulus is going to be our inciting event, right? The input into the system that's going to trigger the receptor or the sensor, same word, sensor. And the sensor is just going to be what is processing the information. Then it's going to send it to the integrator, who's going to control the set point. And in most cases, the integrator is going to be the brain. And then we're going to send it to the effector. And then from there, the effector could be a muscle or a gland. In the case of us, it's probably going to be a muscle, right? We're going to jerk our hand back. And then from there, we're going to have a response, right? Here's another good example that I used in the case of temperature. So recently, we just had this huge uh, Arctic blast that just kind of came across from the west. We saw that temperatures drastically decreased. So, what would have happened if you would have went outside in um, in shirts and a t-shirt whenever it was like 4 degrees Fahrenheit? Well, your body would have sensed that. So the inciting event would have been the temperature decrease, right? That would have been your stimulus. And then the sensors, right, or the receptors would have been the temperature receptors present within your skin. From there, we would send temperature receptors, would send a message to the hypothalamus, which is going to be present in the brain. And the hypothalamus is responsible for setting the control set point. So it's going to say, hey, body, we need to warm up, right? So in order for that to occur, we're going to start to shiver. Shivering is going to increase metabolic rate, which is going to produce heat, which is going to help us warm up. And from there, we may also see something called vasoconstriction, vaso meaning blood, and then constriction meaning just to clamp down. So the blood vessels will clamp down. And it does that in order to conserve heat, right? So again, you have a sensor here detecting the stimulus, and then from there, it sends it to the integrator, and from the integrator, it sends it to the effector, which is going to be the shivering of the muscles. This is another good example here that we have in terms of blood glucose. Blood glucose is um, it can you can either be hyperglycemic or you can be hypoglycemic, right? So, um, glycemia is just another word for glucose. So I like to think of this as the fed and the fasting state. So what do I mean by the fed state? Well, I mean mammal just cooked a big meal at Thanksgiving and you just gorged way too much. So we would say that your body is in the fed state, right? So when that occurs, your blood glucose is going to go up above, shouldn't go above 120, but oftentimes it does. So your body has a built-in response to make sure that blood glucose doesn't get too high. And it's going to do this through the production of a hormone called insulin. And this will be covered in the endocrine section in AMP2. But for purposes right now, I'll just kind of go over briefly what insulin does. So, insulin will be produced by the beta cells of the pancreas. You don't need to know that. All you need to know is that insulin is responsible for lowering blood glucose levels. It takes it up into the liver and stores it as something called glycogen. And it stores it as glycogen because there's going to be a time later on when you're actually not eating. You may be going to ball practice. You may be going down the road and go grocery shopping, and you're hungry, right? You haven't eaten yet. So what does your body do? Well, your body is low, right? Your body is low. You haven't had anything to eat. Your blood glucose is low. So what's it going to do? It produces a hormone called glucagon, and glucagon is produced by the alpha cells here in the pancreatic islets, and glucagon is going to be responsible for glycogenolysis. Glycogen meaning stored glucose, olysis meaning breaking down. So it's going to break down the stored glucose in order to maintain your blood glucose level at um, an appropriate amount within your body. So again, this is just kind of an example that your body has two different types of hormones in order to maintain blood glucose at an appropriate level. So again, you can see that if your body were to shift in um, too high of a blood glucose, that would be bad. If it were to shift and go too low, that would be bad. That's why we have a set of regulatory processes that maintains this blood glucose at an appropriate level, and that's called homeostasis. So we've kind of talked about this condition of equilibrium being homeostasis. So now we're going to discuss one of the ways it does this through it's a cycle of events that we like to call feedback systems. And feedback systems are not very complicated at all, right? You have a reaction that occurs that is going to produce a product. In the case of negative feedback, when you have too much of a product, you're going to tell the reactants to calm down, chill out, stop making that product. That's called negative feedback. The products go back and tell the reactants, hey, we've got enough products, we're good. Positive feedback is going to have some reactants, right? Some things that are being made, and they're going to continuously feed off of each other and make more products. The more product that is made is going to produce more reactants, and it's going to wind up like a vicious cycle. We'll kind of go over this. So, negative feedback. We're going to discuss it first in the concept of stress. Has everybody heard of the stress hormone, cortisol? Well, if you haven't, you'll discuss that later on in endocrine and anatomy and physiology too. So, stress is going to occur. Let's say you've got a big test coming up, and you guys are worried because you've not studied, you feel like you're going to fail, or whatever the stress in your life may be. There's some areas in your brain that is going to produce different effector molecules or effector hormones that are going to wind up producing a product. And we'll go over a few of these today. The hypothalamus, it's just a portion in the brain that is going to produce a hormone called corticotropin releasing hormone, CRH, right here. CRH is going to go and it's going to produce something called ACTH or adrenocorticotropin releasing hormone in the pituitary. CRH produces ACTH within the pituitary, and eventually you have the production of something called cortisol, or it's another word for a glucocorticoid. So I just want you to see that you have a reactant here that is making another reactant right here, and eventually you're going to have a product within cortisol. So, what happens once we have too much cortisol? Our body's way of preventing an excess is by going back and saying, hey, reactants, CRH, chill out, dude. Stop making so much. So, this is called negative feedback, right? Because we're going back and we're saying, hey, calm down. We don't need as much reactants as you're making. When this becomes low, right, and we don't have a production of cortisol, then it's allowed to start um, making CRH and ACTH again, right? But when cortisol's high or glucocorticoids are high, it says, hey, corticotropin releasing hormone, CRH, calm down. It inhibits this CRH production. Um, Positive feedback, a good example here is how a baby feeds, right? So this picture here is going to demonstrate a little bit. So a baby begins suckling at the nipple. This is going to be the stimulus, and it's going to send in a the receptors here are going to send an impulse to the hypothalamus, again, the portion in the brain that's responsible for controlling the set point. And from there, we're going to produce oxytocin. And oxytocin is just going to be a hormone that is responsible for milk ejection, right? It's going to be responsible for what they call milk letdown, milk ejection from the nipple. So again, we have this baby suckling, message is sent to the hypothalamus. We have the production of oxytocin. Oxytocin is a feel-good hormone as well. It also is responsible for milk ejection. So the milk is ejected, and then because the milk is ejected, the baby can continue suckling. So because oxytocin is produced, the baby continues to feed. The baby continues to feed. It continues to go to the hypothalamus um, to release oxytocin. So again, this is just an example of positive feedback. We have one reactant continuing to make a product, and the product is continuing to make the reactant. It's a vicious cycle here. Briefly, we'll discuss the central dogma of molecular biology. And dogma is just a word for belief or ideology. And this is essentially the basis of how we came to be, or how all things are living, or where they come from. It's going to go over some basic principles. And the basic principles were covered in Bio 112, and you probably knew the central dogma before you even knew it. Um, So we'll begin by creating DNA. DNA synthesis occurs. Does anybody remember the enzyme that's responsible for DNA synthesis? Okay, it's DNA polymerase, right? Does anybody remember what DNA is made of? What three components? Phosphate residue, right? Nitrogenous base. Does anybody remember the four? Adenine, cytosine, thymine, and what's the last one? Guanine. Good. And then the other, that's right, deoxyribose. So those are the three components that makes up DNA, and it differs. Or from DNA, we have DNA synthesis, right, through DNA polymerase, and from DNA synthesis, we go to form something called messenger RNA, and this is going to occur through a process called transcription, and we're literally creating a transcript um, using messenger RNA in order to create RNA. RNA is ribonucleic acid. And do you remember how it differs from DNA? Well, you should know that DNA is double stranded, whereas RNA is single stranded. And what's the other difference? RNA uses one um, nitrogenous base that DNA does not. You remember? Try RNA uses uracil instead of thymine, right? So again, DNA is double stranded and it uses thymine, whereas RNA is single stranded and uses uracil, right? So once we have DNA synthesis, right, through DNA polymerase, we then create messenger RNA through transcription. And through transcription, we have messenger RNA. We're then going to undergo translation and then produce RNA and then eventually produce a protein. So here we'll go over some key terms before we get into it. This is DNA or deoxyribonucleic acid. Like we previously said, this was double stranded, consisting of adenine, cysteine, uh, that should be cytosine, not cysteine, cytosine, guanine, and thymine, right? Um, from here, we know that a nucleic acid contains genetic instructions used in the development and functioning of all living organisms. Um, so RNA is a nucleic acid, consists of long chain nucleotide units used during translation to build proteins, like we previously just said. Consists of a single strand, contains adenine, again, cytosine, guanine, and uracil. Messenger RNA, this is just the transcribed message of DNA. Again, this is going to be needed in order to make RNA. Um, Then we have the nucleus. This is where both DNA synthesis and transcription occur. The nucleus is just an organelle present within a cell. The ribosome, remember my Bio 112 class, anytime you hear the word ribosome, you should think of protein synthesis. So these are complexes of RNA and protein that are found in all cells and responsible for translation of messenger RNA into a protein. So you'll see this next picture. It's going to be very important. So once we have proteins, then we're going to begin forming polypeptide chains. This is just going to be an assembly of amino acids that are going to eventually fold into proteins. And then once we have the proteins, the Golgi apparatus is like FedEx. They're responsible for sorting and sending out macromolecules such as proteins and lipids. This is the picture that is going to be very important, especially for examination or quiz purposes. Again, we're going to begin with DNA replication. From here, DNA replication is then going to move through DNA polymerase, right, to form double-stranded DNA. And do you guys remember what the term is for a new strand and an old strand? Semi-conservative. That's very good. Awesome. That's very good. Do you remember the enzyme responsible for unzipping the genes? Helicase. That's awesome. Great job. So from here we have DNA, and again we know this occurs within the nucleus. And from having DNA, we're going to then transcribe the message, right? So we have the notes. We need to get it over onto the computer, and in order to do that, we need to uh, actually that example is not going to work. So let's just do it. Let's just go through it. DNA synthesis is going to occur, right? You have DNA replication. From there, transcription is going to occur. Transcription is going to form something called messenger RNA. That's going to be in between here. Messenger RNA goes through something called post-transcriptional processing to form RNA. RNA, we're now at the ribosome. Because we're at the ribosome, we're then going to undergo translation, right? And then from there, translation is going to form a protein. And then there was another box over here. You'd have the Golgi apparatus, and it's going to sort it and send it to where it needs to go. This is just a kind of like the same thing that's occurring. DNA is occurring here. We have DNA replication. The DNA is going to duplicate. From there, we're going to form messenger RNA through RNA polymerase, right? We have transcription occurring, RNA synthesis occurring. We have the production of RNA. Then from here, we're on the ribosome, right? The ribosome's right here. And then translation is occurring in order to form the protein. So this is a very good picture in order to kind of illustrate that. Um, from here, this is just going into proteins in general, how they're read. How do we know which protein we're going to form? Well, we know that through amino acid residues. And the way this forms, the way this occurs is whenever you have amino acids, remember, which are the building blocks of proteins. These amino acids are going to be coded for different this codon is going to code for different amino acids. So, for example, um, in order to start translation for the protein synthesis to begin, there's only one start codon. I want you guys to know that there's only one start codon that allows translation to begin, and that is AUG. AUG. Um, so from here, translation can begin, and we can begin reading the different nucleotides which are going to be present. And remember, a combination of three nucleotides is called a codon. So let's say that we had AAU, we've got asparagine from AAG, which is lysine, and we could have glycine, GGA. Um, so again, this is just a brief review of things that we've previously covered. This is the building block for the foundation of science, the foundation of who we are as individuals. And if these are not familiar, do not stress out. I understand that these basic science concepts are sometimes difficult, but it's good to have at least a fundamental understanding. So, this will conclude the first introduction lecture. Again, if you guys could watch this prior to coming to class, that would be awesome. So, thank you guys.